And actually, thank you to all three of you because they've been sharing pre-publication data. So a lot of it was really a preview for many people. So it's very kind. So now the goal is really to have a discussion at high level. And because a lot of it is not published yet, we will wait for the publication to have all of the technical details. But do you guys want to share anything on that point before we open to the more general question? No?
It's a secret. It's not. I mean, it's not a secret from the panel from our point of view is you can take any commercially available PCR panel. Well, I'm not going to name the manufacturer we've taken one from, but you do need to fiddle around with the polymerase conditions. Say Joe and Nick have done all the hard work. Need to fiddle around with the polymerase conditions to optimize the amplicons. But then the advantage then is the company has done the balancing for you, so you can then take that PCR panel and stick it straight into the flongle. It's not that complicated. I shouldn't really say that. I want you all to work with me. But actually, as a technique, it's very straightforward. And you'll see it in the paper.
Thanks, Andrew. Do we have questions from the audience?
Hi, Luca Mazzarella from Milan. So, regarding the last talk, so it's not entirely clear to me, however, what is the advantage of sequencing the PCR amplified panels with nanopore as opposed to, whichever other short read sequence? Because your amplicons are small in size, aren't they?
Yeah, I mean, our point is that, so we've tried we do quite a lot of stuff in our lab. We do the TreeSci Liquid 500, another hybridization panel, and the workflow to go from a hybridization panel to result is 5 to 7 days, which is too slow in some of these cases. One of the things that hopefully I perhaps didn't make it as well as right at the beginning is that we can go from the DNA to the result in 8 hours, which is important. You can't achieve that with hybridization. And you can't do that with short read based sequences because you need to run the flow cell for 24, 36 hours to get data. So that's the key advantage to us, and that's why we developed it.
Thanks.
Thanks. We had a question from Anna on the first row.
Thank you for introducing your different methods. Great to see the different modalities being used. When we think about liquid biopsy, we're thinking early detection. Early detection means difficulties with, actually picking up those rare mutations just by default because they're early. And I think this is what we're seeing with current methods that really, they can detect those variants, but they're actually not detecting them early enough at the earlier stages of cancer development. How are you trying to approach that main issue from my perspective with your current methods that you're developing? And that's the question to the whole panel.
I mean, I think in general, the hardest problem really is getting enough DNA for sequencing. So in our case, we've demonstrated that we can start with actually a very small amount of DNA, but the ideal method would be processing a large volume of plasma, get as many DNA molecules as possible, and getting as much throughput. That's more or less a budgeting issue then, right? What is the trade-off between how many reads you need for specific sensitivity and specificity, and how much of a cost analysis that you're willing to put in? Because clearly you can sequence, if you have a lot of DNA from, let's say, 10 mils of plasma, 20 mils of plasma, that's like 2 or 3 blood collection tubes. You can get a lot of DNA, and you can sequence at 30. You can sequence that to 30x on a couple of Promethean flow cells. But the question is, do you really want to do that?
So Caroline, do you have something to add?
I do. From my point of view, so I agree with Billy on the more plasma we have, the more DNA we have, the more likely it is that we're going to detect the mutations that are relevant or of interest. In the case of pediatric cancers, that is obviously not always the case because the plasma volume, the blood volume that we get is very, very small. So we're kind of sort of scratching at a limit of what we can detect. But I think there is a lot of value in being able to read out the methylation data that might be more informative on a read-based level than picking out particular mutations might be. And what we're trying to do at the moment, so obviously we do long-term want to move towards early detection, but another application that is often forgotten about when we talk about early detection is also the disease monitoring and earlier detection of relapse. And I think that's in particular where the application of nanosequencing of cell-free DNA is going to be quite strong and maybe easier to achieve in the nearer future before we then go to true early detection and solving it all. So that's my point of view on this.
Andrew?
I think one of the things we've done up until recently is always relied on mutations to tell us whether somebody's got cancer or not. It's not that easy. The more, like Caroline and Billy have both said, the more plasma you have, the more likely you are to detect them. But what you do notice is a shift in tissue type or tissue phenotype, which can only really be detected epigenetically. So the methods that Billy, Caroline, and we use to detect epigenetic change are probably going to be more important for these early screening assays. So somebody asked about input, like Carolyn says, they use a mil of plasma from the SMPs, and then we're talking about 3 or 4 nanograms of DNA. But one of the things that not just us, but others like Stanford realize is the nanopore kit, it works on femtomolar loading, not quantity. So if the DNA fragments are so short, 150 femtomolar of DNA is, 5 or 10 nanograms of DNA. So that's easily within the reach of most of these pediatric samples, and that's the key to unlocking a lot of these epigenetics tests.
So Billy, on the very low input, do you have some specific comment? I mean, from the preprint, how low you went into quantity of material?
Yeah, so I mean, in the previous published paper and also in the current study that we're doing, we work with a very small amount of plasma, about like half a mil of plasma effectively, that goes forward to sequencing. We did a couple of measurements about what that roughly corresponds to. It's anywhere between, let's say, a couple hundred of picograms of DNA up to like a little bit over 1 nanogram of DNA on average. And that's more than enough for us. But the key trick is that you have to have a very optimized library prep.
Right.
And there's a couple of tricks that we use. It's not that interesting. You just read the paper. It's more about like being very smart about how you make the library, monitoring what your conversion efficiency is. And that's one thing that we're able to do very well that we were able to monitor in the genomedicine paper. And then once you have that, then it's more about productionizing the whole process, because a lot of the follow-up study that we have in our breast cancer cohort, it's about sequencing large numbers of patients. And in that case, you have to have some automation, some good hands, like a good extraction system to do this in volume. That's the hard part.
Thank you. Do we have comments specifically on the early detection before we move on to the next topic? Comments or opinion from the audience? Observations? Specifically? Go ahead.
So I have a question about the DNA methylation. So I guess this is for Carolyn and Billy. Billy, on one of your slides, I think I saw it said that you're querying 400,000 CPGs. So the question is, what is the optimal number of CPGs that you want to be looking at, and what depth of coverage is optimal?
Yeah, so the 400,000 CPGs refers to our feature selection step, where we have a case control sample. We have all the CPGs that were sequenced, and we look for statistically significant differences in the methylation values between our case control cohort. And what falls out is the 400K CPGs. But obviously that changes with the size of your cohort, how deep you sequence and everything. So for us, it's not a specific cutoff, but more to do with after you do your sequencing, you do a statistical test, do the FDR correction, and whatever comes out is what you're looking at for your feature selection.
But it's whole genome, and
it's
the
whole
400,000
genome, yeah.
come out of the analysis.
Yeah, that's correct.
Okay. And what depth of coverage?
We have roughly like a couple million reads per sample, so that roughly translates to also a couple million CPG sites per sample as well.
So, I mean, we have done this as well, and I would add that, like Billy says, there's no optimum amount. So we can see as many as 1 million CPGs in some samples, and some have only got 50,000. Generally, 5 nanograms of DNA will give you about a gigabase, 2 gigabases of sequence data, perhaps more, depending on the sample. So, I understand from the work that Carolyn and Isidro have done that, the classifier doesn't need that much information to pull out methylation data. So it sounds, once again, a bit flippant, but you literally just put in what you've got and see what you get out, and usually that's enough to classify. It certainly saves taking the gallons of plasma off children that used to happen.
Yeah.
Cool. Jeroen?
Yes, thanks a lot, guys. Really awesome. So, comparatively to the mutations and the methylation, how important is the fragmentation pattern? And you look at size, I presume, but do you also, for instance, look at the end motifs of the fragments that you
Yeah.
observe?
So I don't know. Do you want to start or?
Yeah. So your question is like, I think two parts. One is looking at the methylation and also the end motifs, right? So the end motifs we haven't looked at yet. That's a really interesting question. But could be some biases depending on what the end repair step cuts off. So that's more of an open thing. In terms of methylation profile, I think that's in general for me, in my opinion, that can be more informative than just looking at the specific cancer mutations, because methylation is reflective of overall transcriptional programs in a cell, right? That can happen on multiple gene levels and whatever. And so methylation patterns are generally going to be correlated all across the genome. So in general, when you're looking at, let's say, one SNP, let's say KRAS G12D or something like that, or BRAF V600E mutation, that's at one spot. And you want to look at that one spot, that's really hard to do, right? If you only have like 1 nanogram DNA, there's 150 molecules at that one spot that can give you that information. versus if you look at like CPG sites, there's millions of them that could be correlated with each other. So it's much more informative to do that as opposed to just looking at SNPs. But that's my opinion.
Yeah, I pretty much agree. And we do look at the size profiles as well, as you saw on the plot earlier. So I think it's also a very important signal. You can do in silico size selection and other things that will be very informative. Enrich your sequencing data for putative cancer reads, which will be very helpful also for methylation analyses. And of course, there's a lot of work being done on nucleosomal footprinting and trying to infer gene expression. And what we're interested in is then also understanding how this correlates with the methylation signal that we read out from the same data, and expect that it should correlate because it's about gene expression. About the motifs, I think this is going to be quite challenging because what we know so far about the fragment end motifs is from Illumina sequencing data, and the technology is obviously very different to nanopore sequencing data. And the challenge that we're going to have with the nanopore sequencing data, so there's no doubt that we're going to find different motifs in the cancer reads compared to the normal reads. But I think those motifs are going to look very different to the Illumina sequencing motifs that we know. So we have to redefine what we consider cancer-specific fragment end motifs. And the reason for that is that, especially when you start sequencing the DNA, the first few bases are a bit inaccurate, and then you massively depend on the performance of the base caller to remove the adapter sequence. And if you obviously remove the adapter sequence just one base pair too many or one base pair too little, you're just looking at the last 4 base pairs of that fragment, and that's going to change the motifs. So obviously Illumina has other limitations, but I think it's going to be very interesting but quite challenging and not easily comparable to the data that we might already know from previous publications using conventional sequencing approaches. But it's definitely something that we're looking at.
Andrew, any comments on the jagged ends?
It's been so expertly answered. What else could I say? The only other thing I would add about fragment analysis is, there were recent papers in pregnant women as well that showed it's not just about the 175, 300 base pairs. We are detecting long fragments now, and that's the other advantage long read sequencing gives you. I mean, we routinely, in the data we generate for Carolyn and Isidro's group, 1,000 base pair fragments. They do exist in plasma circulating from tumours, and they are tumour specific because they're methylated in a tumour specific way. So these long reads are actually quite informative when it comes to classification, and I think they'll play a more important role and a unique role.
Yeah.
Thanks. We have about a couple minutes left. So do we have any further questions? Yeah.
Hi, it's quite a technical question for Billy. Is that okay?
Yeah.
Yep.
If it's okay for him, yeah.
Yeah, so love your work, love the paper, and particularly love how much detail you've given in the methods. So I work at the vet school up in Edinburgh, and we've been inspired to kind of try out and develop a liquid biopsy for dogs. So do you have any tips or tricks for getting started, particularly as we might move towards kind of kit 14 versus the older kit that you used?
Yeah, so we're actually doing kit 14 on some other studies in the lab right now. I would say if you read the paper, there's a second end repair and A-tailing step. You don't need that anymore, and that's it. Then you can do it on V14.
Thank you.
We do have a technical suggestion as well. I can't say it out loud. Joe knows what it is. There's a certain brand of Eppendorf tube that you shouldn't put. It's not made by Eppendorf. Do not put your plasma in it because it destroys the nanopore. But come and talk to us afterwards. We can't mention it in public because we'll get sued.
All right, thank you very much. So we are running out of time. That being said, you've seen them. They're going to be around. So feel free to walk out to Andrew, Caroline, and Billy. Let's thank them one last time. That was a really great session.